Students, welcome to the session. Uh, in the last session, we had discussed up to the ninth question. In this session, we'll continue with the from the 10th question onwards. See, students, read the question. 10th question given here. Which of the following is an incorrect statement? See, in this given four statements, we need to find out which is incorrect statement. Read the first statement. The nuclear poles act as the passages for proteins and RNA molecules in both the directions between nucleus and cytoplasm. And the second option, the mature sieve tube elements possess a conspicuous nucleus and usual cytoplasmic organelles. Third option, the microbodies are present both in plants and animal cells. Fourth option, perinuclear space forms a barrier between the materials present inside the nucleus and that of the cytoplasm. See, these are the four statements given. Out of these four statements, one is wrong statement. The wrong statement we need to identify. Before identifying, just to see the little explanation. The student here, nuclear pores, which are present on the nuclear membrane. These pores actually regulate the passage of molecules between the nucleus and the cytoplasm. The nucleus and the cytoplasm, permitting some to pass through the membrane but not others. It means membrane always semi-permeable in nature. The building blocks for manufacturing DNA and RNA are some of the materials that are allowed into the nucleus, as are molecules that provide the energy for constructing genetic material. Ribosomal subunits, which are built in nucleosomes, are a prime example of materials that must be allowed to leave the nucleus and enter the cytoplasm. See, nuclear pore complexes in the nuclear envelope. Now you can see this is the nuclear pore. So this nuclear pore will allow the molecules from the cytoplasm to the nucleus. In the same way, when RNA molecules which are synthesized, like mRNA, which is synthesized inside the nucleus, and it can be transported into the cytoplasm. In the same way, the proteins which are synthesized in the cytoplasm can be imported into the nucleus, like export and import by the nuclear pore. It means what? The nuclear pore will allow the movement of proteins and RNA right from inside. It means proteins from outside to nucleus, and then RNA from inside to the cytoplasm. Tell us. Then you see, student, another thing, that is a perinuclear space. This perinuclear space is the luminal space between inner and outer nuclear envelope by layers that separate the nucleus from the cytoplasm. Now see here, this particular space, right? Now see, this is the inner membrane and this is outer membrane of the nucleus. Between these two membranes, the space present, that is called perinuclear space, right? This space actually separates the materials between the cytoplasm and the nucleus, right? So we'll see another statement. Sieve tubes. See student here, the function of sieve tubes are believed to be controlled by companion cells in mature sieve tube, right? Nucleus in generally absent in the sieve tube. Sieve tube there is no nucleus. It contains a lining layer of cytoplasm only, and it means it does not have nucleus and also cytoplasmic cell organelles. Cytoplasmic organelles, cell organelles also absent, right? Okay. Now see here the microbodies. Microbodies. See, actually, these microbodies they found in both the cells. Both the cells. What are actually microbodies? Microbodies are usually considered to be a single membrane enclosed organelles approximately 0.2 to 1.5 micrometer in diameter. Now you see here, student, Christian D. Dewey et al. Isolated microbodies from rat liver cell. It means from animal, right? From animal. And see, in plants, the microbodies were first isolated from the castor bean seedlings by Bredenbach and beavers, and from spinach leaves by Tolbert et al. It means what? The microbodies are present in both plant cell as well as animal cell. Right? Okay, now we'll see the options, the statements given. Here, the 4 statements. See the statement number 1. The nuclear pores act as passages for proteins and RNA molecules in both the directions between the nucleus and cytoplasm. This option is correct. It's not incorrect. And see the second option, mature sieve tube elements possess a conspicuous nucleus. It means they do not have student option 2. The mature sieve tube elements possess conspicuous nucleus and usual cytoplasmic organelles. Here, it does not have the nucleus and cytoplasmic organelles. It means the statement number 2 is wrong statement. It means incorrect statement. In the third statement, microbodies are present both in plant and animal cells. Correct. The perinuclear space forms a barrier between the materials present inside the nucleus and the cytoplasm under the cytoplasm. So this is also correct. So incorrect statement is the second statement. So second option is the correct one. Right? This is the correct option for this question. Now see the 11th question. Student, 11th question given here. Match the list 1 with the list 2. List 1, lenticels. A is lenticels, B is cambium, C is secondary cortex, and D is cork. And then list 2, phallogen, sovereign deposition, exchange of gases, and a phallodam. Now see here, little explanation. See, in generally the lenticels helps, right? Exchange of gases. Now see here, lenticels are the structures formed for allowing the escape of carbon dioxide and also for entering of oxygen. It means exchange of gases. So cork consists mostly of suberin material. Suberin is a waxy substance found in higher plants. Now see student here, this is here phellum. This component here, this is the phellum or cork. And see here the cork cambium. The cork cambium is here phallogen, cork cambium, and the secondary cortex is phallodam. Right? Now once again we'll try to match the options. Now see the lenticels, exchange of gases, right? This is third option. Caucambium, that is a phallogen. 1. Secondary cortex, pallidum. 4. Cork, sovereign deposition. 2. See here A3, B1 and C4 and D2. See here this third option is the correct option for this question, right? Okay. Then see another question, 12th question. The amount of nutrients such as the carbon, nitrogen, phosphorus, and calcium present in the soil at any given time is referred as. The options given: standing crop, climax community, standing state. See student here, there is a confusion between standing crop and standing state, but remaining these two terms are not at all related with that. There is a confusion between standing crop and standing state. Now we'll see that their explanation. The student here, standing crop. What is standing crop? The amount of organic matter in a given area at specific time that we can call as a standing crop. Otherwise, the standing crop is the amount of biomass present in an ecosystem. That is standing crop. Regarding this particular agricultural field, now how much of biomass is present in this area? That you can say that is standing crop. Now see, the standing state is amount of inorganic nutrients found in an ecosystem. Amount of inorganic nutrients present in an ecosystem. For example, as we've seen that previously in the options, like carbon, nitrogen, phosphorus, calcium, sodium, and these amounts in a particular ecosystem, in generally soil, that can be called as a standing state. Now read the options. See, standing crop regarding the organic biomass, and the climax not at all related. Then climax community and standing state is the amount of nutrients such as carbon, nitrogen, phosphorus, and calcium present in the soil at any given time. So correct option is a fourth one. Now see another other question. Right. Okay. See the 13th question. The term used for transfer of pollen grains from anthers of one plant to stigma of a different plant which during pollination brings genetically different types of pollen grains to stigma is. See student here, options given: the clistogamy, xenogamy, chaetnogamy, chasmogamy. Right? Before we are going to find the correct option, we'll see the different types of pollinations. See here, the autogamy. It means the pollen transfer within this single flower, right? The anther of flower into the stigma of the same flower. And chaetnogamy, within the same plant, between two different flowers. The transfer of pollen grains between the two different flowers within the same plant. The plant is same here, right? Now see, in the xenogamy, the transfer of pollen grains from one plant to the another plant within the same species, right? That is xenogamy. In generally, the xenogamy brings the genetic variations, the different genetic material containing pollen grains, right? Now, when you talk about the chasmogamy and clistogamy, those two are a type of autogamy. autogamy pollination. Now see the clistogamy. These are actually closed flowers. So here there is no genetic variations, and deithnogamy also not that much genetic variation, and chasmogamy also not that much genetic variation. And in the xenogamy, it is what cross pollination, where it brings genetic variations. Now this is the correct option for this question. Now see the 14th question. Which of the following stages of meiosis involves division of centromere? Division of centromere. Centromere division occurs in which stage of the meiosis? Options given: telophase 1, metaphase 1, metaphase 2, anaphase 2. See, among these four options, we have to find out the correct option in which stage the centromere divides. Just will compare, student, all the, actually that is at the anaphase stage. We'll see in all the three different types of anaphases we'll discuss. In case of mitosis, just at the anaphase division, at anaphase phase, what happens? Centromere divides. In generally before anaphase, means chromosome will be like this. Chromosome is like this. This is what at centromere region. See, during anaphase, the centromere get breaks, right? With the help of enzymes, and the spindle fiber pulls the two daughter chromosomes into two different poles, right? That actually happens in the anaphase of mitosis. But when it comes to the meiosis, there are two anaphases. The one anaphase, that is one, and then second anaphase 2 in second meiotic division. See, in anaphase 1, exactly what happens? Centromere is not getting divided. Instead of centromere division, the entire chromosome is moving into the pole, right? Is being pulled by the spindle fibers into the pole. In anaphase 1, there is no centromere division. When you see the anaphase 2, centromere is getting divided, and then two daughter chromosomes moving into two different poles, right? So the centromere division in meiosis takes place in the anaphase 2, not in the anaphase 1. Now see the options. So telophase 2, not at all centromere division, and metaphase 1, the arrangement of the chromosomes at the middle of the cell. The same way metaphase 2 also same. And anaphase 2, exactly the centromere gets division, and then chromosome, this chromosome separates into two daughter chromosomes, right? So the correct option for this fourth. Now see the one more question, that is 15th question. Which of the following is correct sequence of steps in a PCR, polymerase chain reaction? The options given: annealing, denaturation, extension. Then denaturation, annealing, extension. Denaturation, extension, annealing. Extension, denaturation, annealing. Right? Among these four options, there is the one correct option that we have to identify. We will go for the little explanation. See, student, here the image. During PCR, the first step is that the double-stranded DNA should become single-stranded, right? That the first step is called denaturation. Double-stranded DNA becomes single-stranded, right? After the denaturation, the next step is the primer binding to the DNA, right? That is called annealing. The next step is annealing. So primer will go and bind to the complementary sequences on the DNA. After annealing, the third step is the extension. So annealing is over, and then the elongation of the chain, the polynucleotide chain will take place, right? That is extension. So the first step is denaturation, then second step is annealing, the third step is extension. Now you can see, student, here the PCR mission, and also here a PCR mission is there. And see here PCR wells, and see the PCR tube is here, nucleotides, and then primer, DNA sample, tag polymerase, and the PCR buffer. Now see the question. See here, first step is the denaturation. See, double-stranded DNA becomes single-stranded. Second step is primer binding, annealing. Then next thought is elongation of the